So, since we are starting with our revision series now, so our first thing is going to be chapter number 5, the fundamental unit of life. And this is going to be a revision lecture, so we'll try to cover it up within 20 minutes or 25 minutes, not more than that. And this is going to be first half of the chapter. So, next half of the chapter will be covered in another video. Okay, let's start with the revision. So the first thing we have in the chapter is about discovery of cell. So we have to understand the discovery of cell in a timeline. Okay, so how the timeline is going to be like timeline of events. So first on the list, we are going to have Robert Hooke. Okay, Robert Hooke is a scientist who discovered the cell for the very first time, okay, in 1665. But the point was that he was having a self-designed microscope, which means it was a primitive microscope. What is the meaning of primitive microscope now? He was having a very basic microscope. Okay, so he couldn't do much with that microscope. He only took the cork cells. Now, what are the cork cells? You know the bark of the stem, right? We have got the bark, which is the outermost covering of a tree. Okay, those cells are known as the cork cells. Like the bark of the stem is made up of cork cells. Okay, now these cells are dead cells. So now what is the problem with the dead cell? Obviously, it is not going to have nucleus. We cannot see all those things which should be there inside the cell. Okay, so it was a dead cell that he discovered, and he named the structure that he saw. What was the structure? He called them as a honeycomb structure. What is the meaning of honeycomb structure now? It was looking like this to him. Okay, so he said that, oh, it is looking like a honeycomb structure. So we can simply say that it is a honeycomb structure with some dead cells in it. And then he also coined the term cell. Okay, where did he get this term from? He took the word from a Latin word which means a little room. Okay, so we have got everything about Robert Hooke now, that in 1665 he got a self-designed microscope, and that was a primitive microscope. Obviously, it was very basic, and then he used cork cells, which means the bark of the stem, and then along with that he discovered the dead cells. Okay, and he named the structure as a honeycomb structure, and he coined the term cell. So these are some important things which they can ask in the exam. Coming to the second person, who's Leeuwenhoek. Okay, Leeuwenhoek came in 1674, and he was having improved version of microscope. Okay, so because he was having improved version of microscope, so it was comparatively easier for him to do any discovery. So he discovered the living cell. Now this is very, very important that we should be able to understand the difference between a living cell and a dead cell. Who discovered the living cell and who discovered the dead cell? In exams, there can be MCQs in which they can ask you who discovered the living cell. So we should be having this thing in our mind. It was Leeuwenhoek. Okay, dead cell was discovered by Robert Hooke. So what did he use again? He used an improved microscope, and he actually discovered the cell within a pond water. Okay, now in pond water, what kind of cell can we have? We can have so many microorganisms in that, right? There will be so many microorganisms, and those microorganisms can be unicellular also. So he just took the microorganisms from the pond water and he discovered the cell within it. So he was the first person to discover the living cell. Now, coming to the third person, he was Robert Brown. This is very, very important. Please understand this thing. I myself, when I was in school, I did this mistake. Okay, in one of my exams, I wrote that Robert Brown discovered the cell. Okay, because these names are very similar. We have Robert Hooke and then we have Robert Brown. Robert Hooke discovered the cell. Remember this thing. He discovered the cell. Okay, and Robert Brown. In Brown, we have N. All right, in Brown, we have got N. So N means nucleus. Robert Brown discovered nucleus inside the cell. It is very, very important that you are able to differentiate between Robert Hooke and Robert Brown in the exams. Okay, so we have got Robert Brown in 1831 who discovered the cell. Coming to the fourth person, he was Purkinje. Okay, Purkinje in 1839 he discovered the protoplasm. What is a protoplasm now? Protoplasm is going to be like the cytoplasm of the cell, and when you take cytoplasm along with the cell membrane, because cell membrane, what is the another name of cell membrane? Plasma membrane. So plasma membrane along with the cytoplasm is known as the protoplasm. So protoplasm was something which was discovered by Purkinje. in 1839. Are years important? No, they are not. Okay, it's just that I have given you the timeline, so I have included all the years. Coming to the fifth person now. In the fifth, we can have two people together, Schleden and Schwann. Okay, now what was their discovery? They gave us a cell theory. Okay, what was written in the cell theory? They simply said, okay, one of the two, Schleden and Schwann, one of the two was a zoologist and another one was a botanist. So they did separate studies. That is why we are having two different years. Okay, one Schleden, who discovered same things in year 1838, and Schwann discovered the same things in year 1839. So they gave us the cell theory now. In the cell theory, what they have written, they said that all the animals and plants are made up of cell. Okay, and the plants are going to have the cell wall, animal cells are not going to have the cell wall. This was also included in their theory, and they gave us a cell theory. But there was something missing in the cell theory, which was that how are we getting these cells? We understand this thing, like the bodies are made up of cell, but how are these cells there? Like, what is producing the cell? So that was a question which was still there and which was not answered by that time. Then comes another person, Rudolf Virchow. He came in 1855 and he gave us the modified version of cell theory. What was there in the modified version? He said that the cells are coming from the pre-existing cell. Now see, you have the answer to your question. Like, who is producing cell? What is that one thing which is producing cell? So we have the answer to it now. Cell is producing another cell. Cell is dividing to produce another cell. So he gave this one, we can say a quote, okay, and it was omnis cellula e cellula, which means what? The cells are coming from pre-existing cell. Okay, you understood till here? Like, what are we talking about? We are talking about the modified version of cell theory, in which we have a person who was Rudolf Virchow. He gave us this theory and he gave the statement omnis cellula e cellula, which means the cells are coming from pre-existing cell. Okay, now we can come to the last part of the timeline, which is 1940. In year 1940, we have the discovery of electron microscope. The best discovery, because right now, today also when you go to the laboratory, you're going to use the electron microscope only there. Okay, so electron microscope, when it came, after that, it led to so many discoveries within the cell only. Because with the help of electron microscope, you can look at the minute structures also. So that is why in 1940 we got the electron microscope, and after that, this research and the studies are still going on. We have got so many discoveries after that. Coming to the second part, which is going to be classification on the basis of number of cell. Okay, most important thing, whatever I have included in the notes, first of all, they are handwritten notes, as you can see. And second thing is, whatever I have included in the notes, it is very, very important from the point of view of exam. Okay, like every single thing can come in the exam. So I have given you the differences also properly, because in the exam, if they ask you the difference, they will be asking you difference for minimum of 2 marks, right? So in 2 marks, you cannot write down just 2 points. This will not work, right? So you have to at least give 4 points. So even if it comes for 2 marks, then I have included all the 4 points, okay, in the differences part as well. So coming to the classification on basis of number of cells, how many types of organisms do we have? We have unicellular and then we have multicellular. What is the difference? Very basic, very simple thing. Unicellular, single celled. Multicellular, multiple cells. Unicellular, one cell performs all the function. Think about it. If you have only one person in the family, obviously that person is going to do all the things, right? In the same manner, if you have just one cell in the body, that cell has all the burden now. It has to perform all the functions. Coming to the multicellular, different cells, different functions. Like the skin cell can go for protection, eye cells can go for optical activity. Then we have hair cells. Hair cells are also going to have some of the other activity, which is mainly protection again. So we have got multiple cells, and multiple cells have got multiple functions to perform. Okay. Then coming to third point, no division of labor. What is the meaning of division of labor now? Division of labor means that the work is divided. Okay, for example, you look in a family. Okay. In a family, we are going to have multiple members. All the members are providing to the family in one or the other way. Okay, everyone is earning, everyone is providing everything. So it is like division of labor. All the work is not done by one person, right? So we can say that there is, like, a proper division of labor is there. Everyone is performing specific function to provide to the family. Okay, same case is going to be there in the multicellular organism. Different cells have got different functions to perform, so they their functions respectively, and that is how an organism is going to survive. Correct. In the unicellular organism, we don't have that privilege. In the unicellular organism, it's just no division of labor, which means one cell is going to do all the functions now. Okay. Coming to the examples, we have amoeba, paramecium in cellular organism, but when you talk about multicellular organism, we have plants and animals. So I hope you have understood till here, like classification on the basis of number of cells. I have seen this multiple times coming in TYQs. So please memorize this thing, okay? Coming to star points now. What are the star points for today's lecture? That shape, okay, these are some of the points which are not included in NCRT, but the thing is they have come in the exams in some of the PYQs, so I have included them over here. Coming to star points, yeah. So shape of cell depends on functions they perform. This is important. Okay, like how can we say that this particular cell is going to have a particular shape? Because it depends upon the function they perform. Okay, it is just about what kind of function they are, similar sort of a shape. For example, all the nerve cells, their work is to connect the body in terms of electrical impulses or nerve impulses, correct? So if they have to transmit the nerve impulses, they have to be long enough, right? So we have the longest cell, which is a nerve cell. So they have a proper shape, they have a proper size because they have a proper function to perform. So, like we can say, the function goes hand in hand with the structure, or vice versa. Like structure goes hand in hand with the function. Then we have another star point, which is pseudopodia. What is pseudopodia now? Pseudopodia is a type of movement. What is a like pseudopodia? We can say it is a movement which is used by amoeba. Do we have pseudopodia in our body? Yes, we do have. Okay, not a movement, sorry. It is not going to be a movement. Movement is diapedesis. Pseudopodia, pseudo means false, and podia means feet. Okay, so pseudopodia actually means false feet, which is there in amoeba. Amoeba structure looks something like this. Okay, now the thing is that amoeba can contract itself and it can relax itself as well. So these, what you are looking at right now, are jelly-like feet that amoeba has, but they are false. It can convert itself into this thing. So now the feet are gone. Now the feet are gone. Okay. So that is why we say that pseudopodia is false feet, which is a characteristic feature of amoeba. And even in our body, we have something similar known as white blood cells. Okay. White blood cells can show pseudopodia as well. So you got to know what is pseudopodia now, false feet. Like it is, we can say when amoeba requires feet, it is going to have it. When it doesn't require it, it is going to remove it. So just like that, a false feet kind of a thing. Then a very, very important question, which is there in NCRT also, that why cell is known as structural unit of life. Now, usually students are very, very confused between the structural unit of life and the functional unit of life. They think that both of them are same things, though they are not. So, structural unit of life, if I talk about, so we can say that why we call cell as a structural unit of life, because millions of cells, just think about it. When you are going to construct a house, you are going to use so many bricks. Okay, so many bricks are going to assemble together to form a house. In the similar manner, we have got millions of cells which are assembling together so that they can form an organism. Okay, now the thing is that these are the minute structures. You cannot go beyond cell. Like cell is the simplest version. Cells are assembling together and then they are giving us this entire organism. So that is why we say that it is a structural unit of life. I have given a very proper answer on the screen as well, so you can note it down if you want. That millions of cells assemble to form an organism. All structures in an organism is the assembly of cells only. So that is why we call it as a structural unit of life. Okay. Coming to the functional unit of life, why do we call it as functional unit of life? Functional unit means like it is capable of performing different functions. Okay, if you look at your body only, we have skin cell, which is going to go for secretion of sebum, which is going to protect our body. Then we have heart cell, which is going to pump the blood, which is known as cardiac cell also. Then we have stomach cell, which is responsible for secreting digestive juices also. It is responsible for digestion also. So you can see this thing that in our body only, we have multiple cells, and multiple cells have multiple functions to perform. Correct. So that is why we call it as the functional unit of life. Like if a cell is not performing a function, then how a tissue will be performing a function? Think about it, because tissue is ultimately a group of cell only. Correct. So if a cell is failing at performing its function, then tissue as a whole cannot do anything. Right. So it is just like if a brick stops, being so strong, okay, it just breaks down. All right, the entire structure of the house is going to break down after that. Okay, it'll be shattered. So the same thing goes in the case of body of the organism also. A cell has to perform a function, so there is division of labor. Each and every cell has different function to perform, and that is why we call it as the functional unit of life. So I hope everything is understood till here. If you have a doubt, you can ask. Okay. So we can come to now the next topic, which is the structural organization of cells. So what is the structure of cell, what it looks like? Obviously, we will not be dealing with the cell organelles in this lecture. We'll try to cover it up in the next lecture. So for now, we can just deal with structural organization of cell, which includes cell membrane. After cell membrane, we have nucleus, and then we have cytoplasm. What is cell membrane, also known as plasma membrane? Let's get into that. So firstly, these points are very, very important. So I'll give you time to note it down. You just note these points down, okay? So plasma membrane. In the plasma membrane, we can say that it is the outermost covering of the cell. Why have I mentioned over here animal cell only? Because in plant cell we don't have that, right? Um, in the plant cell, you remember I told you this thing that in the plant cell, the outermost covering is the cell wall. Okay, we don't have the outermost covering as the plasma membrane. So the plasma membrane is the outermost covering in the animal cell. Very important point. It is a living layer. Okay, this is a very important difference between a cell wall and the plasma membrane. Cell wall is a dead layer, okay, whereas plasma membrane is a living layer. So I'll give you the differences between plasma membrane and the cell wall also in the next lecture. For now, plasma membrane is a living layer. Third point is it is made up of lipid and protein. Very important question. Okay, see, sometimes in the exam we get the, decision making, not decision making, I can say, like those kind of questions which are not known by everyone in the class. Okay, like everyone doesn't know the answer to the question. But if you know the answer to the question, then you can be like, some of those students who can score good. So you should be able to know all the answers. Okay, you should be able to have all the points in your mind while you're writing down about any topic. So we have got third point, which is the lipid and protein. So it is going to be made up of lipid and protein. Fourth point is going to be selectively permeable in nature. What is the meaning of selectively permeable in nature again? That it is going to allow only certain substances to pass through it. What is the meaning of that now? Plasma membrane is so selective in nature that it is going to work as a checkpoint. Okay, so what kind of a checkpoint now? It is going to look at the molecules. If they are gaseous molecule or water molecule, it will allow the water molecule and gaseous molecule to pass through it. But if it is any kind of other molecule, then it will ask to pay some amount, and that amount is going to be in the form of energy. Okay, so selectively permeable in nature means what? That it will allow only certain substances through freely. Okay, then you'll be having a question that, ma'am, how is it possible? Because we have seen so many molecules, glucose molecule is there, proteins are there, so many molecules are there which are entering into the cell, but they have to pay an amount. Okay, so like we can say selectively permeable means partiality. That cell membrane is little partial in nature. It will only allow like gases and water molecules to pass through it freely. Then we have a difference, which is in between diffusion and osmosis. What is difference between diffusion and osmosis? In both of them, the molecules are moving from a region of their higher concentration to a region of their lower concentration only. But the thing is that in the case of diffusion, there will be no interference of a selectively permeable membrane. Like it's gonna be just molecules moving in the air or moving in whatever state they are moving, right? They whatever state they want to be in, and then there will be no semi-permeable membrane interfering in their movement. Okay. In the osmosis, we have the movement, but the movement is going to be through semi-permeable membrane. Okay. Coming to diffusion, how is it going to help the animals and plant in exchange of gases? Even in our body, if I talk about in our body also, the diffusion is helping in exchange of gases. Okay, coming to osmosis, osmosis helps in intake of water. Then we have different types of solution, which is going to be the last topic for today. So what are the different types of solutions? Hypotonic, isotonic, hypertonic. What is hypotonic? Hypotonic is a kind of solution in, okay, see, these solutions, when we talk about these solutions, they are in relative manner. What is the meaning of relative manner? We cannot talk about a solution if we do not have a, like we can say, a cell in it. Okay. Like I can say a solution is hypotonic, but it is hypotonic in respect to the cell. Okay, like if I have a cell, let's say, and cell has got more solute in it. Okay, then if I'm placing it in a solution and solution has got less solute, then I can say the solution is hypotonic with respect to the cell. Okay, so it is a relative concept. Like I cannot take any solution to be hypotonic without having something else to compare it with. Okay, so if let's say this is our cell. Okay, and the blue molecules, obviously, they are going to be water molecules only. So these are water molecules, and these are solute molecules. Okay, so you can see in the first case, we are having cell, and cell is having more solute than the solution placed outside it. So this solution will be named as a hypotonic solution. Now, hypo means less. Okay, tonic means tonicity, or tonic means you can say less solute. Okay, so we have got less solute in this solution in comparison to the cell. Please remember that it is a relative concept. Okay, you cannot just look at a solution and call it as hypotonic unless and until you have something to compare it with. Okay, so in this type of a solution, if a cell is placed, then cell can undergo endosmosis, which means the water molecule will start moving from outside the cell to inside the cell, and cell will become turgid. What is the meaning of turgid again? Swollen. Okay, it will be swelled up. Then we have isotonic solution. Iso means same. Okay, same tonicity. What is the meaning of same tonicity now? Same amount of solute and same amount of water outside and inside the cell. Like cell has also got the same number of water molecules, and outside the cell in the solution also we have same number of water molecules. So in this case, when water molecule is not having any concentration gradient, then there will be no movement. Okay, so we are going to have no net movement over there, and cell will remain flaccid. Flaccid means in its normal condition. No endosmosis, no exosmosis. Then we have the last type of solution, which is going to be hypertonic, in which hyper means over. Again means tonicity, which is going to be solute. So it will be having more solute than the cell. Okay, so if a solution is having more solute than the cell, then think about it, what is going to happen? If you're having more solute than the cell, a solution, then the water from the cell, since it has to move from region of higher concentration to lower concentration, so it'll be moving outside the cell now. Okay, and when it is moving outside the cell will become plasmolysed. Exosmosis will take place and it will become plasmolysed. What is the meaning of becoming plasmolysed now? It will lose its protoplasm. Cell membrane will be gone, cytoplasm will be gone. Okay, so cell is eventually going to die. We know this thing that cytoplasm is the like fluid part of the cell. So it is helping in survive. It is helping in cell to survive. Okay, so if it is gone, obviously the cell will die. So we have got all the three types of solution also now. Then we have next topic, which is going to be cell wall, then nucleus, and all these topics we are going to cover in the next lecture. And I hope you have understood the lecture. After this, we can have 15 minutes for the doubt session. Okay. So you can ask your doubts also from me. Okay, so just go through all the notes first. Okay, if you want to note down anything, just let me know. I'll take you to that portion. Just note down everything and then let me know if you have got any doubts. We'll discuss that.